So, hello and good morning, everybody, and welcome to day 2 of our Smart Lab Exchange R&D event. My name is Daniela Jensen, and I will be your host today. And I'm responsible for product marketing at Biovia Dassault Systèmes. I'm now 20 years in the industry of lab informatics and actually attending Smart Lab since day one. And I would actually like to start today by thanking our speakers from yesterday, the panelists, the leaders of the roundtables, and also our charming Claire Annette, who did guide us through the day. It was a great event yesterday with many different topics, and I hope you enjoyed it. What I found interesting that across the 5 presentations from industry and vendors, the 3 roundtables, 2 panel discussions that we had, a topic that came up a lot was data standardization. I was a little bit surprised about it because if you are in R&D, you might be focusing more on innovation, maybe on latest technology. This was my expectation. But data standardization seems to be a big topic. And I think that's really because it's the basics. It's the basics that enable us to do all the cool things in R&D. So, but we also touched on other topics. We discussed data analytics, AI, and machine learning in different areas. It was collaboration and its impact or its change during the pandemic, then automation, integration, connectivity, data management. And in one of the roundtables, we even discussed the customer-vendor relationship in the lab space. So I hope you enjoyed it and that you did get something out of it that really helps you to improve and change your work, your daily work in the lab. Today we have a little bit more overarching topic. So it's about the lab of the future, lab 4.0, digital transformation, and but also topics that are more basic like cloud or fair data. So we will start with 2 presentations about connected smart labs by Abhishek from Amgen, then another presentation about data harmonization in the cloud by Mike from TetraScience, followed by business meetings, a roundtable session, and another round of business meetings. For the roundtable, you have 2 options. So one is the future of fair data management, led by Whitney and Kellen from Collaborative Drug Discovery, and bridging the automation gap through data integration, led by Nicola from GSK. Then we will meet again for a presentation about digital transformation by Hayden from Merck, a panel discussion about Lab 4.0 and metadata capture, led by Magdalena from LabTwin, and a last presentation about the SILA standard. Also, that means going back to the topic of standardization, and this given by Burkhard, who is also a board member of SILA. So I think it's good if you stay until the end at 5:15, because we will be doing a draw for a giveaway of 100 euros Amazon voucher. And so make sure you stick around and you get a chance to win it. So like at day 1, we will be allocating time for questions at the end of each session. And I've seen yesterday there were actually not too many questions. And therefore, I would really like to encourage you to ask, because it's your opportunity, your chance to learn more from the experts about your specific interests. So don't be shy, ask questions. So with this, we move to the first session. And this is held by Abhishek Mathur. He is working as a director of global research at Amgen and leads externalization operations for Amgen's therapeutic discovery organization. Besides Amgen, he has also worked at Regeneron and Catalent, where he held various CMC functions while supporting product development and drug commercialization. His primary background and interests span across CMC, operations, and structured innovation. And he said he's passionately driven by technology-led innovation. So I think this will be really an inspiring session. So the floor is yours, Abhishek.
Thank you, Daniela, and very good morning, good afternoon, good evening to the wide variety of audience that we have today. I don't have a nice revolution backdrop as Daniela had, but I'm equally passionate about it. And thanks to the team for inviting me for this great conference, great meeting, and always happy to talk about or bring out the passion around the smart labs of the future. And the future is here, as I always call it. So in today's discussion, today's presentation, I'll be talking about this optimized, automated, and connected labs that have been driving the industry primarily in biopharma sector. Can you guys hear me okay? Anila or Daniela, can you confirm?
Yeah, Abhishek, we can hear you fine.
Great, thank you. So before delving into the meat of things, it's important to understand how the industry has evolved. And yes, in history, we read about industrial revolution. Industry has changed quite a bit from James Watt steam engine to mass production in late 18th century and early 19th century. And then it took a long time to graduate from that, even though innovation continued to happen on newer things, newer novel stuff. But with the advent of computers in early 60s and 70s, that has, in conjunction with the automation, with the machine, the turnkey robotics, I think it has started to change and take a bigger leap. Then there was again novel stuff continued to build up, but when it really got into its log phase of growth, as I call it, is in last 10 to 15 years, where we are not just using computers and automation, but we are integrating it with smart and autonomous systems and utilizing data and machine learning, which was totally unheard of 30, 40 years back. So this is a great time to be in industry, and sure, there will be 5.0 and further on, but I think the way industry is moving and evolving, it's a great time to do a lot more stuff with the resources that we have. Moving on to the healthcare industry, which is not untouched by this revolution, it is transforming rapidly. And based on one of the surveys by one of the financial groups, healthcare innovation will reach the warp speed in this decade. And this is calling it out and by a big margin, because healthcare, if you think about it, it's not more than 50, 60 years old. And the way we have evolved, it has been transformational to lives of people, to lives of patients, to the society as a whole. And in this paradigm, when we have artificial intelligence, machine learning, we have automation and robotics, we have cloud, where data can reside, where people can collaborate, we have data analytics and visualization mechanisms, and then there's something blockchain. I think these things are really catapulting healthcare industry in a totally new paradigm at such a fast pace. So this is again very not just interesting and exciting, but this is game changing for the society as a whole. And being a biopharma person, I'm extremely committed to bringing these medicines to patients and changing bring a change to their quality of life. And this is if I think about what is happening, if I see what is happening, if I use what is at my disposal today, it has been an immense immense motivation to continue working on this. So rapid advances in modern biopharma within healthcare have created opportunities to make these medicines faster, cheaper, and smartly. No more we are putting one table at a time, waiting for 10 years to have a biologics molecule come out. No more we are spending a couple of $1 billion for making a commercial ready molecule. No more we are failing 99.8% of our pipeline molecules just because they could not make it till the end. So speed, scale, and throughput have become really essential. We want a drug in 3 years now. COVID was a great example where this vaccine came out within a year span, and this showcases what industry can do with the tools that they have today, and how the world has changed or has started to change once this COVID vaccine came through. And we don't just have one vaccine; we have at least 7 or 8 vaccines that are already commercialized and lot more that are under approval stage. So this is this should excite all of us to the same degree. And if we don't use these tools, I think that the price is much higher to pay, not just in terms of money, but in terms of the society. Society, what we can make a change to it, and it will suffer if we don't. So in next few slides, I'm presenting you an exciting opportunity, Lab of the Future, which allows us to fast track our innovation at core. I'll go over how synergy of science, engineering, and design elements are amalgamating together and making things happen at such a rapid pace and with such a high quality. We have a smart and flexible integration of technologies. Before we were using it in silos, but now they can be integrated to achieve the quote unquote warp speed and the high throughput that we desire to make our scientific decisions. To enable these things, we have right robotics and automation now available that eliminate the manual task and allow scientists to think more about the application part, more about the design part. So that is amazing. The physical work, people used to be called golden pair of hands and all these things. That was great, but now with this automation at our disposal, every assay, every experiment can be of high quality and deliver the right things, while our scientists can focus on designing it. And all this is fueled by our digital empowerment with data at our disposal. We can make data-driven decisions. We don't have to rely on repeating the experiment that was produced, reproduced from a different lab altogether. So this is all coming together nicely, and it is really driving labs of the future, and the future is here, as I always say. A quick snapshot into history. So tools that at our disposal continue to emerge in last century, in 20th century. But 1981 was a key turnkey year where Zymark introduced turnkey robotics and automation that labs can use. And these automation could be used, it was like a liquid handler, it could be used, first of all, mass produced, and it could be used in different lab settings in different industries. But it took almost 3 decades to get out of what I call liquid handler syndrome. Even in last decade and the decade previous, I've been across different companies. I work for really big pharma. I work for a CRO. Most of these places, the thought almost stopped at calling liquid handler as their technology. And everybody felt proud of it, that they have 2 or 3 liquid handlers and they can do things faster. But what beyond that? How we can change the game and be a step up faster? Nobody was thinking in a structured way at that point. So early 2010s, that's when the advent of artificial intelligence, collaborative robots, workstations that can be integrated to create integrated workflows, big data, software, visualization systems, all these suddenly come to the fore and had a commercial value, and people understood how they can integrate. So last decade was really instrumental in changing the whole game in biopharma industry. Now, and especially last 5 years have been tremendous. You see, this technology revolution is not happening not just in Bay Area or limited to United States. It is everywhere, in Europe, in Asia. Currently, I'm in India right now, and the way the startups are coming up on extremely difficult things they are trying to tackle, it's amazing to see. Same thing happening in China. So the whole world has realized its importance, and the technology that has been there at our disposal, now people have started to use it in a more tangible manner, integrate these technologies in a more tangible manner to make big things happen. So some of these tools I already enumerated, like these novel technologies, automation, which is a big component. It speeds things up really quick. It provides the quality. There's innovation happening in lab operations. The workflows are getting more integrated and end-to-end. The strategy is evolving in that direction. We have R&D informatic platforms and the live IT systems. Companies now have big IT organization that have a data strategy at their disposal, how they can not just store data or manage data or retrieve data, but also allow data to make data-driven decisions. And artificial intelligence and machine learning has really projected in a totally different orbit now. So what is the structure of a modern R&D lab? So it has basically 3 pillars. Every lab has a certain workflow process, so it's designed to enable an efficient execution. This workflow is enabled by the use of automation and robotics nowadays. Modern R&D labs, earlier it used to be manual, but now we have machines that can do the work for us and software that can store data. And then it is an informatics platform. So these platforms don't only store the data that gets split at different points of the workflow, but also try to allows us to take data-driven decisions along the way as the workflow is progressing. So these 3 pillars have really enabled modern R&D labs to do much more with much less and fast track drug development process by order of magnitude and making it cheaper in the process as well. I've talked about some of these productivity enablers, but reiterating that reliability. These robotics has really provided us a reliability and quality. Integrated workflows has enabled the speed and scale. It has reduced the cost because now we don't need multiple people to do a small job. Machine can do job quickly, faster, in a much more cost-efficient way. These modular setups can be put together to do a variety of different experiments now. So flexibility exists without changing the plans and making sure we have these 10 people told to do things differently every single time. Machines are much easier to operate. You can quickly program them. You can have dedicated engineers to do that. Now we have better control. We can sit in the comfort of our home and still manage, still execute the experiment. And then the cloud databases, the data residing in cloud has enabled collaboration all across. However, with all this excitement brewing, with all these startups and different technologies building up, there's still a lot of current challenges. We are not mature yet. In no way we are mature. We are just started on this journey, and individual technologies may be maturing more than others, but how it will come together, how it will become a commonplace across industry, it is still yet to happen. So we are still in a maturation stage. Secondly, some scientists are wired to have control on their experiment. With cloud coming in, with automation performing the actual experiment, with different groups having different ideas of how to execute an experiment, there's a cultural acceptance and cross-functional alignment that is a challenge, especially for big pharmas. And an overarching strategy needs to be put together, and a proper focused leadership should be there to make things happen in a more tangible, in a more productive way. Further, because of the variety of systems emerging all across, there's a lack of global standards. They still cannot be integrated seamlessly. You need a dedicated team of engineers, computer engineers to program them. These integration of these labs require a lot of investment of time, money, and effort. And then with that comes the business risk. What if it doesn't come through? What if an alternative is faster and much more reliable? And what if the partner that they're working with closes, they shuts down? So investment is a big deal, but again, it is coming down over time. Building flexibility to run experiments of wide variety of nature, again, is challenging. It's design intensive. So it requires, again, dedicated workforce to build that flexibility in. Digital infrastructure, informatic platforms should co-evolve with data strategy also. So they need to be in rhyme. They need to be in sync with each other. And then I got about the business risk. What if it doesn't work? What if it doesn't work as we project it? So because of all these challenges, an overarching focused dedicated leadership is extremely important for any organization to make things work. Further, there are a bunch of different external partners that are working on a variety of technologies and providing services that can really help drive down the costs, pilot things out, and thereby mitigate the business risk associated with building something internally. So there are ways how different companies are operating and mitigating some of these possible challenges and risks. But again, this will continue to evolve as we move forward. There will be some key players that will continue to emerge, and at some point, I'm really expecting somewhere down this decade, down the line, we can have an end-to-end drug development process from discovery through commercialization. So this will be an amazing feat for biopharma altogether. So in line with that, the smart lab market is set to grow 18% CAGR year-over-year growth rate. Again, it's one of the surveys that says that, but we are already seeing this unfolding. So in next few slides, I'll cover some of the examples, but just to give you a good glimpse of what are the possibilities, some of the possibilities. This is not comprehensive by any means. So this example is from a Ginkgo Bioworks, who recently got a great deal in IPO and a $16 billion deal. And there's a biofoundry model. This model is called biofoundry model. I'm not sure if you can see the top part. I cannot remove it. But it says biofoundry model, which is a DBTL model. You design, build, test, and learn, and it's an iterative process. And this is all integrated. And once you have optimized the condition, then you manufacture at scale. So this is all integrated, all end-to-end integration with very little manual intervention. So this is the biofoundry concept by itself. It is adopted from the standard foundries, in manufacturing sector that people have. The idea is everything is end-to-end without any manual intervention, and it includes robotics, it includes machine learning to make data-driven decisions, and it is all integrated within the same location, same brick and mortar location. And this is one of the examples where Ginkgo has really made a difference to an industry already. They work very closely with one of the commercial COVID vaccine manufacturer, and they identified these neutralizing bodies at scale. The process was standard. You design the lead antibodies, then high throughput DNA synthesis, followed by antibody expression on a miniaturized scale, followed by screening, and then they integrated a beacon-based system for B cell screening and identifying the lead candidates. It was followed by all sort of analytics that happened. So this process is all integrated end to end, and they had very successfully in a very short time identified the candidates, and out of which one has become a commercial vaccine already. So this is a great example where the smart labs can really make a difference, and they have started to make a difference. It's not just a vision anymore. It has been realized repeatedly in past 4 or 5 years. Another example, I can remove it now. Another example of a smart lab is a robotic cloud lab model. So a scientist can sit down in his chair at home and can operate, can open a software on his computer and can run an experiment end to end. So Emerald Cloud Labs has that model where they train scientists on very basic scripting, and scientists then run all those experiments while sitting in the comfort of their home or office. So this has been great because when COVID happened, labs got closed and companies were wondering how we can run our operations. But having these kind of options can really make things work. They can also make things work by allowing the scientists to monitor things from home and make sure that everything is going on well. It is not to infringe on one's work-life balance, but it is to just to make sure that within 8 to 5 or whatever the times the person is working, it can be done efficiently and it can be operated really quickly. Amgen has collaborated with Stratios regarding this cloud lab for medicine, for chemistry, discovery chemistry work. And we have, our scientists, which have been marked by, let's say, our scientist has been working and operating the lab through cloud platform, and they have this actual platform in their brick and mortar lab. So scientists are operating it, and not just scientists from one side, but scientists from multiple sites are operating. And after site A has done their stuff, then the output goes to another work cell, which is operated by site B, and then it goes to the another work cell in the same location, which is operated by site C. So this has been pretty successful collaboration with Stratios on the medicinal chemistry work, and we are trying to look into biologics as well. And Emerald and Ginkgo are working on their biologics portfolio and developing these work cells or their workstations to support different biologics pertaining workflows. Important thing. So after these examples, it's important to talk about data and not forget it, because that has really changed the whole landscape in past 5 years. I think that was one thing that has allowed integration of all these software, all these automation, and made it into a seamless end-to-end process. We have different research software that can monitor and control the specific process. We have research software forever. We have had data management platforms like electronic notebooks or LIMS platform to store the data and use it for documentation purposes in the past. What has really emerged lately are the informatic platforms like Benchling, then there is Dotomatics. So there are a lot of platforms, and there are some other indigenous ones as well that companies are developing or co-developing with existing skeleton platforms. And then in conjunction with this, the artificial intelligence or machine learning initiatives to allow that stored data to be used in a more tangible way and feedback into the process for next step. I think this has really closed the loop and made that process sustainable and with an intelligence almost of a scientist. So this is very important, and to that effect, Amgen, I mean, this slide talks about how Amgen has invested in its data management, data platforms. So I won't go into every single block in here, but this shows the complexity as to how, this is example for protein reagent generation process. So how cloning, expression, analytics, and purification, all these have been integrated with different research software and discovery platforms, and have resulted in a very seamless working ecosystem. Sure, we need a good IT workforce to support this, but I think it is very well worth it, as it has really sped up our drug development workflow. Besides this drug development, it can the smart labs can be very modular in their approaches. It can be used. Integrated workflows can be used for protein designs, protein expression, drug discovery and optimization, in cell therapy space, cell line development and engineering, metabolic engineering, next generation modalities, small molecules, and whatnot. So the idea is to put machines together, integrate them, and then let them do the magic. So a variety of applications this can be used for. Earlier, we did not have options. we could find one option, one single company is working on something really cool, and we try to find it. But the best part of today is we have a lot of options. We need to identify what fits to our internal processes, internal workflows, and then just grab the opportunity. Because time is now to act and build. And I really want to make sure and put the point across that it's important for biopharma industry as a whole to adopt this model vastly and rapidly, because this industry, we who belong to this industry, we owe faster medicines to the patients who are waiting to get these drugs and improve their quality of life. Thank you. I think a lot of people to thank for, primarily Amgen's leadership and our discovery team, and some of our external partners who we have been talking to, and I stole some of the slides from. Thank you.
Thank you so much, Abhishek. Thank you. This was nice to see that actually the lab of the future is here today, right? With all the automation, connectivity, and real end-to-end integration and remote lab execution. I really find that very cool. So we have questions, and so the first one that I see here is, what are some of the ways, like tasks, decision making, that AI and machine learning is being used for lab work?
So I give an example, like protein synthesis, and let's have a membrane protein, which is a very complex protein, and you need to have multiple iterations, optimization. So you create a design and identify, for example, the design of experiment, and you rely on the past data sets that you have for similar domains. And that can provide you what kind of conditions would work appropriately for creating a new domain for this protein. So past data is important. You know, what we store as humans, and hey, for making this helical structure, and with a very small pocket, we use these conditions, and we used this sequence that really helped. So we store in our mind or our notebooks, but AI can retrieve that information very rapidly. It can search for hundreds of similar structures and give us the best conditions and maybe best sequences at a micro scale, and allow and feed into our machine that can make that protein with that sequence, or that domain with that sequence. So it is very powerful, and a lot of companies have come up with this kind of models. AlphaFold, I don't know if Tim has heard about it. In next 2 to 3 years, they will be they'll be publishing all the structures of all the proteins that are known to mankind. Now, they have not been experimentally verified, but their technology has been more than 99% accurate on the test set, and that's what they are banking on to say, okay, those thousands of these proteins, we expect that 99% of those structures will be accurate. So this is powerful, especially for a structural biology scenario, and this can be applied for a variety of different lab work.
Yeah. Okay, thank you. We have another question. We don't have much time anymore, but I really want to read this. It's standard. So Dale Charlton is asking, standards is a real issue. Many lab instruments still controlled by PC software running on PCs. So I see the average R&D lab has dozens of PCs and rarely integrated into a network. And if integrated, a huge IT project, and few labs outside of pharma have these resources. So how to democratize the technology? How do labs know when to jump into the technology? Is it a VHS versus Betamax syndrome for me? Unless we can develop lab standards like CELA 2, what do panelists think about CELA 2 and bringing standards into labs connectivity and lab of the future?
I think industry is maturing, but it is not matured yet. And putting standards right now can really jeopardize the innovation that is happening, because some of the systems may not make it because of the standards. That's always a possibility. Having said that, having a small IT team, computer software programmers, that can really help in creating patches that will allow different software, different machines to talk to each other. In fact, there are some companies who are providing these services. Their business model is to provide these IT services who can integrate these systems all together. So it's important, first of all, to see for your business, for your company's organization's business model, is it worth spending on this yet or not? And at that point, when you agree that yeah, if you need a scale on these things and you have to keep making these data-driven decisions as you move forward, at that point, it's a very wise decision to invest in the software programmers who can make things work. Start on a smaller scale and then build it further. These standards will always come through over time, but my only guesswork is because everything is growing in every different corner of the world, it will take some time before they come up with a common set of standards, and they need to be a consortium and tons of different logistical challenges that will exist when they come up. And on the USB disk question, I think industry has moved very, very quickly onto the cloud platform. So USB, at Tangent, for example, USB is not even permitted to be used for data transfer.
Yeah. Okay. Fantastic. So thank you so much, Abhishek. And we are running a little bit late, but only a little bit. But it was exciting enough to spend a little bit more time here. And now we are moving to the presentation from Mike Tarselli about accelerating digital transformation and AI machine learning applications in life sciences R&D through data harmonization in the cloud. So Mike Tarselli is Chief Scientific Officer at Tetra Science, and he ensures impact of the Tetra R&D data cloud through content leadership, training, the partner network strategy, and building their scientific culture. Previously, Mike was the scientific director at SLAS, a global professional society dedicated to lab automation, and an associate director at Novartis, building an external collaboration networking platform. He's currently on the board of some associations like the Stoya Alliance, the UMass Amherst College of Natural Sciences, and is a member of the American Chemical Society and others. So I would like to hand over to you, Mike.
Thank you very much, Daniela, and that's a very kind introduction you made. Thank you to the organizers for having us. Thank you to everybody for listening for the next few minutes while we explore what cloud might mean for your scientific data and business. So, all right, my title, a little bit long, but I'll go through it with you: Accelerating Digital Transformation and Artificial Intelligence Machine Learning Applications in Life Sciences R&D through Data Harmonization in the Cloud. And if I may I have everybody mute only because I'm getting some feedback on the line? Thank you. All right, so very briefly, we've already done the introduction, so I'll go past that. We'll talk a little bit about how data will impact your AI machine learning in the life sciences. We'll talk about laying the foundation for this AI machine learning. We'll talk about how R&D data harmonization and integrations are core to this journey, and then we'll obviously leave some time for questions and answers. So we've met me. I'm literally wearing the same shirt. We can skip over this part. I'd love, in fact, to start with this quote. This is from Dell EMC, large provider in the managed service and storage and compute space. Healthcare and life science organizations have seen an explosive growth, 878% since 2015. And this was quoted in June of 2021. What does that mean? Nobody wants to do the back of the envelope math, so I'll say it's about a 50% growth every single year for 6 years straight. And in reality, it hasn't been that consistent 50%. It was a little incremental in 2015, 2016, and it's taken off recently. So it's probably more like 80 or 90% growth recently. And we're not even talking about the growth in terms of just compute. We're talking about storage as well. We're definitely past exabytes now in terms of what we're talking about storing here, and in terms of what people are doing with their scientific data. More is being generated, more detailed data for precision medicine, for synthetic biology, for neural networks and AI to start up from. So what is the strategy of your pharma, biotech, academic institution, government agency for this explosion of data and the contextualization of that data? A great example was just published. I mean, I couldn't have made this up any better. And actually, Abhishek, thank you very much for sort of passing the baton for this talk, because this is great. This is AlphaFold 2, right? As you probably are all aware, DeepMind, which is a Google AI machine learning research firm that spun out into London, won the CASP challenge 3 years ago by predicting a couple of different human proteins very quickly. And now this time they've refined the algorithm. AlphaFold 2 can now predict 24,000, I believe it is, human proteins to within experimental accuracy of their final solution structures. And why is this important, and why am I talking about this? First, this is the downstream impact of what you could have if you had your data in one place, rationally organized, curated, fair, and able to be mined for this type of impact. And second, this is cloud on top of cloud on top of cloud. Where did the test data come from? Well, Uniprot, hosted on an AWS cloud. Where did some of the initial data come from to refine the algorithm for the first challenge? Well, PDB, Protein Data Bank, also hosted on cloud. Where did the researchers make their engine available? On a cloud. Really what you're seeing is that the cloud is this democratizing space that allows the sort of impact and engine to power the future of biomedical research. All right, I think we're going to stop for our first poll. So if the organizer could please chime in for our first question to engage the audience.
Okay, Mike, I'm going to bring this up now. So you've got the poll coming up. Where is your organization on the path to digitalization and replatforming to the cloud? You've got some options there as well. Fully embrace digitalization, have replatformed all our workflows data to the cloud. In the beginning to middle stages of implementing digitization at my organization. At the conceptualization and planning stages of digitalization, yet to initiate, not part of our business activity. So I'll leave that up for just another 30 seconds if everyone could vote.
Yeah, A is you are a digital pro. D is you just don't know much about the cloud and you're not really ready for it.
Okay, Mike, I'm going to bring up those responses for you now.
Excellent. All right, so just about everybody says, hey, look, we are thinking about digitalization and we're in the middle of it. However, we're not finished with it. Very few have not initiated, which is great. So this is a very learned audience that's already on their path to the digital journey. But definitely the two main things, as you can see, are B and C, which are, in the beginning to middle stages or at the conceptualization. So there's still time to think through this, and there's still time to plan your cloud journey. And maybe some of what we say today can help influence that. All right, moving along. What do you need if you want to do artificial intelligence and machine learning? Given the short amount of time I have to speak with you today, I'm not going to solve for you how you lay out a neural network, how you exactly curate your data. We'll only be able to speak to some of the salient features one will need in order to go down this journey. So needs, fair, right? Findable, accessible, interoperable, reproducible. This was defined about 6 years ago by an international consortium of scientific librarians. And actually, it's very important inside of any institution that does science, right? Where can the data be seen? Where can you get access to them so you can do your own mining or work or interpretation? Will they work in multiple different models? And can you repeat that analysis over and over with the same conditions and specifications such that you produce the same result? All right, full data lifecycle. When data gets generated, does it just get pushed off someplace, never to be seen again? Does it get stuck in one format and perhaps never, ever interconverted or utilized? I've seen some scare studies in which people say something like 70 to 80% of the data ever generated by an organization is touched by one set of eyes and never seen again. I'm not sure if I subscribe to that. However, I will say that people don't traditionally say, okay, when will the data be up? When will it be valid and usable? And when should we retire, sunset, or delete that data? All right, should we ever delete that data? These are questions your organization will need to ask itself. The 5 V's. What are the 5 V's? Volume, how much? Velocity, how fast? Veracity, how true? Variety, how diverse? And value, what's the worth? So do you actually have these 5 V's mapped out of your data streams? Do you know why you have certain data streams? Do you trust these data streams? Can you keep up with the volume and the velocity of these? So I know we talked a little bit about needs as challenges, but let's also pivot into the challenges of having an on-prem and sort of fragile data ecosystem. First, silos inevitably build up, right? Your biological data sets for your imaging and perhaps your imaging on this particular program might actually only be in one specific DB, only accessible by a handful of your researchers. Well, what if data scientists in preclinical want to see that? What if manufacturing actually could learn some lessons from the cell culturing you did and want to scale them up for bioprocessing, but they can't get after your data? What is the impact of that to having data having to be regenerated, for example? Failure and fragile poem connections. If somebody starts up with a new instrument or a new technique, invariably they'll say, actually, I just need the data coming off of this to go to this file share that goes to this visualization, and then later downstream, I'll ask my data team to do one model or visualization. That's fine for your process at your time. But again, that inhibits the reproducibility and interoperability, the two IR of FAIR, from being able to be done. And in general, you're going to inherit a maintenance burden. As you bring in a new connection or technique, the human sort of intention is to take that and say, okay, I'll just build another fragile point-to-point integration and another and another. And eventually you have these spaghetti strands of links between systems that don't help anyone, and it's a difficult map to maintain. And then limited or no data science capabilities. Many organizations start the journey and say, we're going to generate data, we're going to do lots of experiments, but then they don't have anyone to look backwards and say, what did we do? Why were we doing it? And consider the data in totality. So the strategy you might want to adopt if you were going to start digitalization today, or if you were in the conceptualization phases, as 37% of you said you were, you'd want an open and vendor neutral approach, because you're probably going to have more than one vendor's software, machinery, instrumentation. You probably want cloud first. As Abhishek just sagely pointed out, they're working with Stratios, right? They're working with several other cloud solutions that allow you to have this fair approach to your data and scale dynamically to accommodate these 5 Vs. And then compliance. You're going to want to ensure that that cloud solution you adopt is going to be ready for things like GXP, ISO 9001, SOC 2, CIS, anything you would need in order to ensure that that environment were safe for the data that's eventually going to impact patient medications and therapeutics. So when you replatform to this cloud, let's assume that cloud is in your future and it's in your near future. Let's say it's a 1 to 3 year journey out for your firm or business. You're going to want an R&D focused cloud, right? Something where scientists have touched its design, because if you just have technical firms from Silicon Valley descend upon this directly, they may not be able to keep the scientific integrity and the workflow cohesiveness. They may not understand why an NMR or a mass spec develop different data sets, or why this particular field or parameter in this cellular assay has to be maintained, or if this can be changed. You really need some scientific expertise behind that. You need a data-centric approach. You shouldn't have to worry about which software, which instrument, which et cetera actually produce that. And you shouldn't have to worry about the source or target systems. You should be able to preserve the context of the data itself as it transits. Think about that as the central asset, and to expand what the worth of that is to your firm. You want a cloud native solution, right? We're showing a cloud in the middle, obviously, but you want, because of this fair access, this 5V scaling, and self-healing, high availability, you want the ability, just like you would with ride sharing or online retail or checking on your taxes online, you want the same thing about the reliability and accessibility of your firm's data. And then finally, you want openness. Ideally, when your business changes or when your research programs change, let's say you pivot hard to protein degradation or to DNA encoded libraries or some downstream omics we haven't even construed. How will you then be able to reconnect your data sources and imagine the re-networking of your systems? Will it be point-to-point where someone will have to physically move and connect systems, or can you plug and play? So I think that leads us into our second poll. So organizer, if you could please put it up. All right, I guess I'll read it. What percentage of your time is spent wrangling or curating data? Ergo, building, testing, maintaining integrations, looking for reports and data, file transfers, manual data entry, machine learning training set, etc. So how long do you spend noodling with your data and physically hand transcribing or typing? Is it A, less than 10%, B, 10 to 40%, C, 40 to 70%, or D, worst case, over 70% of your day? Do you perhaps spend 6 hours or more of your working shift integrating and wrangling data?
Hey, Mike, I'm going to bring that up now. Sorry, I was on mute there a moment ago talking to myself, but I'll let another 10 seconds and then I'll bring up the results for you. Okie dokie, coming up now.
Oh boy. All right. So we run this poll a lot. And when we run it, we are often surprised by the results. So a plurality of you, over 83% of you, spend over 10% of your day. And since the stats are edging towards the 40 to 70% and higher, over 50% of you say that, that tells me that if you were working a solid 8-hour working day, many of you listening to and answering this poll are spending 4 hours or more taking data, moving data, uploading data, changing data, formatting, curating. Gosh, that's a lot of time. And my guess is that when you were in graduate school or training in your research positions or training in your IT positions, you weren't trained specifically to curate data. So you are actually doing something that will take you away from doing your experimental pursuits. All right, so let's talk about what we could possibly do to help solve that. Network scale. You want more integrations than anyone, right? You want to be able to take on any instrument, any software, anything that comes, right? Even things we haven't developed yet. This is actually the TetraScience, our company, our roadmap for what we've turned on and what we've enabled for scientists to date. You can say we dipped our toe in the water in 2018, very, very few native integrations, and it's sort of followed this power law curve. Very, very fortunately, and actually because of application of strategy, getting ahead of this, we now sit at around, this is actually out of date. By this month, we're actually at around 160 native integrations with about 100 on our roadmap. And now we have, we're projecting around 220 integrations with another 200 on our roadmap by next year. What does that mean? What's an integration? We are defining integration as end-to-end data management, high integrity, with harmonization in the cloud, with scientific contextualization carried all the way across, with the ability to be reused, reinterpreted, with a full audit trail and governance back. So you are able to link to a mass spec, a cell counter, a DSC, a light scattering instrument, an FTIR, a Raman, a bioprocessing unit, whatever. And in addition, many different scientific software, such as been mentioned before, Benchling, Dotmatics, IDBS, whatever have you, in a highly reusable way that you can apply to any scientific workflow. So what's an example of this? Recently, we released a blog post in which we and our partners Benchling allowed us to see if a researcher, for example, was making a Benchling query, say they want to start an experiment or request for an assay, it might transit through our cloud, through what we call our connector. This is an API, web-enabled way to transit data and requests from Benchling over to us. We then ferry that through application programming interfaces or APIs or software development kits to an instrument of choice, right? A couple of them are represented here. This other scientist says, hey, thank you for that order. I'll help you run this. And in some cases, we can actually issue the command directly to the instrument, skipping the scientist entirely. We then harvest that data through an agent or connector, agent if it's an SDK, connector if it's an API. We bring it in regardless of format, and we can bring it in a very highly structured way, like an Excel database or a tabular format, and a very unstructured way, or rather hidden structured way, like a vendor binary or a database. And then finally, we can move it through the Benchling connector back to where it belonged, right, to its origination point. So all of this can happen in an automated way without anybody manually moving data, transcribing data, downloading, interpreting data, et cetera. And it lands in Benchling auto-magically, almost as if you didn't know it had been done. This is an example in which my colleague Joel sets up an example in Benchling, very granularly, sends it over here, moves it from our cloud environment through SDC, Smart Line Data Cockpit, a familiar bioprocessing application. It moves it into a Roche SEDEX, it's a bioanalyzer, helps you count cells and apply properties to them. Joel, again, sees that data. It gets taken in by the SDC connector, moved up through our cloud and back to Benchling. And Joel basically sits back at his desk, or thinks about another experiment while he waits for this cycle to complete and then looks at his results natively in Benchling. Really, it's almost an irony of this space. If we do our job well, we are nearly invisible. However, we are facilitating great advancements to both the instrument data collection and harmonization and to Benchling itself for allowing you to see the data in the source of truth. How are we doing this? One way is through this harmonization that we keep talking about, a format into which we are taking the data out of the setX and we are putting it in JSON, JavaScript Object Notation. This is similar to approaches taken by other firms, but importantly, it's not a standard. It's not a standard, but it is a way of uniting data from multiple different instrument models of one family or of multiple different of an instrument from different vendors or different types and putting it in one human readable, easily accessed, indexed form. So later when your data scientists look at these tables, actually when your scientists look at these, they'll look and say, oh, okay, I get it. You ran this kind of sample on this kind of device. Here's where the data is, okay. But your data scientists see this and they say, whoa, you have an indexed tree of everything that's ever been done in this experiment. And it's all easily accessible on a cloud, in a data lake. I can get it all, I can model it all. And that allows you to set up things in visualizations, in Spotfire, Streamlit, R, in minutes to days, as opposed to weeks to months. It's very quick. These visualizations that I'm showing you now, this is a runtime and sort of accessibility operations layer. This is a chromatogram overlay. These were created on massive data sets from our data lake, probably in a couple of hours by a researcher, who had seen the data once, had looked at some common headers and basically said, okay, here's what I want to do. And it was a very simple exercise because the data had been pre-curated, pre-harmonized, and indexed. All right, so again, we want to accelerate your time to insight, right? Through harmonization and prepared R&D, we're going to allow you to get data that's ready for AI and machine learning. We want automagic integration. Ideally, you shouldn't have to have manual intervention. You should be able to click a button in an ELN, a LIMS, or something, and you should just see data flowing and populating without you having to interfere. Your systems are always up to date because we maintain them that way, and there are web processes going on in the background that allow this. We want to pioneer new technology, right? If you decide you want to bring on a new sequencer or a new mass spec or a new any kind of device, something that hasn't even been developed yet, you can do so if we have a connector. You just search for the connector, you say, okay, we're ready to go, and it's plug and play. Matter of hours to maybe a day or two, as opposed to months to develop something new in point-to-point land. And finally, you can expose hidden insights. Well, maybe your harmonized data allows you to see very subtle things you would have missed otherwise, like whether or not a specific column affects a substrate during purification, or if there's a cell culture that performs the best because you can see the cell culture across an entire project over time. I think we have time for one more poll. So organizer, if you could please bring that up.
So this one is, what percentage of your data can you access today? Less than 10%, 10 to 40%, 40 to 70, or more than 70%.
And remember, this isn't effort like the last time. This is what can you see if you went searching.
Okay, then I'm going to give that another 5 seconds. A couple more still participating. All right, Mike, answers coming up now.
Gosh, I mean, it's predictable, I suppose, but it's a little bit unfortunate, right? Only 15% of you said you could see more than 70% of your data landscape. Again, the plurality of you, 63%, said you could see under 40% of your data. That means that if you are sitting in a QC/QA function, where ideally your function is to inspect the data and the veracity of it, to protect people downstream, right? To ensure clear submission to regulatory authorities and to protect patient safety, you can unfortunately only see under 40% of your data. How will you then make good decisions? How will you make better models? That's a good question, something we should definitely keep in mind. So to that end, again, TetraScience's mission, we accelerate discovery, we improve human life, right? We want to reduce your costs, increase your research efficiency. We want to enable this collaboration and break down data silos wherever we may. We want to accelerate your time to insight and ideally future-proof your data strategy. As Yogi Berra said, it's very tough to make predictions, especially about the future. But we can at least say that if you don't have your data organized in a place and accessible, it's very unlikely you're going to be prepared for what comes in 5 years. And then the sort of compulsory one slide of who are we and why should you do business with us. We enable centralized, harmonized, and actionable experimental data through our R&D data cloud. We are the first R&D data cloud for life sciences and are solely devoted there. We do not provide cloud services in other verticals or other domains at this time. We are simply life sciences, pharma all the way. We are combined expertise in life sciences R&D with cloud native mindsets, data science proficiency. We hire a lot of scientists and a lot of world-class data architects and engineers, and they pair together to design something that's an additive product. Former researchers from Harvard and MIT are our co-founders. We have over 160 native integrations. We support work at CROs, CDMOs, ELN and LIMS companies, data science instruments, and we are currently deployed in 15 of the top 30 global pharmas. And shown here, these are not stock photos. This is literally the view from our office. This is literally the view of our platform. This is literally a lab we have worked with, and this is literally part of our executive staff. So we are trying to be as transparent, open, and helpful as we can be. All right, and I think I've left just a few minutes for Q&A, but I pass it back to Daniela. Thank you.
Thank you for your transparency, Mike, and for explaining us, the value of the cloud, of harmonized, centralized data for R&D, and I think beyond. So, yeah, and we have a question here. It is, how does the Tetra Cloud address compliance like 21 CFR Part 11 and GXP?
Sure, great question. 21 CFR Part 11, for those of you who are perhaps not on the regulatory side, is about electronic records, right? It's about specific audit trail, it's about signing authority, it's about having a software development process that is easily explainable and controlled, and it's about having appropriate training for those who would access your platform or your data. So we have addressed all of those in multiple recent customer and partner audits. And in those, we have set up, for example, electronic signing authority of our policies. We have a full audit trail to go back through your data, any actor, any time, any particular file in your lake. We have full training records available upon audit. And we have a full software development lifecycle that is getting better refined and automated every day. That again has been pressure tested recently. So we are well on that path. We are GXP ready for you.
Yeah, I think that's an important aspect. So
oh yes,
I have another question. Yeah, because in development and quality, that's absolutely basic. So yeah, but it's good to hear that you're addressing this. And then we have another question about how does the Tetra data platform harmonize heterogeneous experimental data?
Right. So we move things into these common formats. Again, we've spoken to them a little. This is the JSON format. It's actually called Intermediate Data Schema, or IDS. What we do is we look at what are commonalities, common data properties, if you will, to various different vendors or data source format representations of experiments. It's a lot of words. What I mean to say is many cars look like cars, right? Even though they may have different paint colors or windshields or aftermarket details, but they're all still cars. They will still have tires. They will still have an engine. They will still have, a certain kind of construction, doors, right? We have that same kind of reduction almost to a genericization that allows you to say, all right, if it is a car, it's going to have these general properties. And then we map that to what that particular vendor needs or wants. And then we add that sort of extra 10% spice of, oh, they also carry through the name of who ran it, or the timestamp, or a temperature property or something. Let's harvest that as well. So we not only surface the subtle metadata, but we also make that generic block so that you can compare that against a different manufacturer.
Yeah, that's important. Okay. We have a couple more questions, but I think we have still time for at least one. So it seems that people that take
choose the best
wanted to ask questions. Okay, okay, let's go for that. How many bidirectional integrations are readily available today, and what future integrations is Tetra Science planning?
Oh gosh, bidirectional. Let's unpack that term a teeny tiny bit. That means something where you start at one point and the data flows eventually back to that same point. So bidirectional traditionally starts with an electronic lab notebook or a LIMS, but could also be done with a manufacturing execution system, an LES. Lots of different systems can kick off an order or request and then be fulfilled. And we help to ferry that data back because we inherit or listen for those commands to come out of that initial originating system. We move them over to the instrument because we have all those connectors, right? And then we bring that data back in this harmonized way and publish it, if you will, over to the originating system. So it's a little strange because you as a scientist might only just be sitting in your IDBSE workbook, right? And saying, okay, I'm waiting for my data to appear. You don't see us operating behind the scenes, but your data, when it comes back, is the right data and has been handled correctly, and you have more data than you thought coming back from the instrument. That's bidirectional. We support that with a variety of things today, IDBS, Dotmatics, PerkinElmer Signals, Benchling, several others. And then our future integrations, where are we going? Just like everyone in this space, I think we're seeing just so much depth across the R&D cascade, right? We want to remind people that R&D also means D for development. So you have just as many physical property checks and preclinical things you have to do and QC/QA on really intense mass spec analyses, for example, than you do research instrumentation. So we're finding many calls for that, many calls for hardness testers and disintegrators and manufacturing control systems, equally almost as we are to systems that control omics or sequencing. So we're sort of doing these parallel tracks to what do people need in D, development, and what do people need in R, research.
Okay, that makes sense. Okay, so we are actually even 1 minute over, but yeah, but thank you so much